Welcome everyone to the BACE event on epigenomics in cell-free DNA and liquid biopsy, and welcome to BACE as well, especially if it's your first time. We are very glad to have you with us today. Our mission is to bring together diverse professionals who have interest in epigenomics, and we do that through scientific and networking events. And we meet to share knowledge and to help create opportunities in epigenomics. We've existed for about 13 months now. We have over 350 members, and we, if you count the event we have today, we've organized 6 larger events, seminar events like the one today. And in between, we also have so-called Chromatin coffees where we meet to learn more about each other and to discuss in a more casual setting. So without further delay, I would like to introduce our first speaker today, Nathan Boley, who is the co-founder and CEO of Ravel Biotechnology. And he will tell us about the cell-free DNA fragmentation patterns and how they reveal chromatin structure. Nathan, thank you so much for joining us tonight. And if you're there, please feel free to take over. Thank you.
Yeah, thanks, Monica. And yeah, thanks for inviting me to speak. I'm really excited to talk about this. So I've been working in this field for a long time now. I think I know a lot of people in the crowd have been working on sort of functional genomics and understanding how we can use accessibility patterns to infer, the structure and function of the cell. And so for most of my career in academia, it was using assays like DNase and ATAC-seq to really understand and characterize chromatin structure using accessibility patterns. And a few years back, it came out that if you look at cell-free DNA, that a lot of the things that people have been designing experiments for over the years are happening naturally inside of the cells. And so I decided to start a company around this, and our company is focused on analyzing cell-free DNA and using the fragmentation and accessibility patterns to infer the chromatin structure. So as I'm sure you guys know, the bad part about starting a company is that a lot of the work that you do is proprietary and you can't really discuss, but there's a lot that's in the public domain and a lot of interesting scientific knowledge. And so I'm going to give a review of sort of, where we're at in the field and sort of show how you can analyze cell-free DNA and understand chromatin structure, and give a really brief update at the end about how we've used that for early disease detection. So please feel free to interrupt me. I think it's a pretty small focus group, and I'd love to sort of make this more of a discussion than a monologue about what we're doing. So like I said, we're Ravel Biotechnology, and we're focused on using accessibility patterns to understand different types of functional elements. And a lot of the sort of pieces I'm going to present are from this review, Zakowski et al. 2016 at the bottom. It does a really good job, in my opinion, of overviewing sort of the field of fragmentomics and how we can use different types of fragmentation patterns to infer accessibility. And so just as sort of a way of orienting ourselves, there's really a few different types of elements that you can identify using accessibility patterns. And this isn't limited to cell-free DNA. This is the same sort of analysis that I'm sure most of the group has done with ATAC-seq or DNase-seq. And that's really looking at a few different things. So looking at the really short fraction that corresponds to pieces that are bound to transcription factors, looking at nucleosome depletion. So that's when you have different protein complexes that pull out a nucleosome so that things like POL2 can bind. Subnucleosome fractions, these are little fragments of DNA as the nucleosomes are being repositioned. And of course, nucleosome position and histone modifications that are associated with different types of cellular processes. Most of the work that we do is using V-plots. And by a nod of heads, can I see how many people have used a V-plot or an analysis on them? All right, I'm getting like a half and half, basically. So the idea behind a V plot's pretty simple. So the idea is that there's really two things that matter when you're looking at a DNA fragment as it relates to accessibility. There's the parts of the genome that it spans, so where the starts and the ends are, and then there's the length of the fragment. And so if you look at, and sorry if this is basic for a lot of people, but just to orient everyone, if you look at, sort of a typical example, you have some nucleosomes in the linker here bound up in heterochromatin, and this naked DNA that spans between the nucleosomes, before the cell does anything, that's all intact. But as the cell dies, undergoes apoptosis, necrosis, and then sheds itself into the blood supply in the case of cell-free DNA, all of that accessible or naked DNA is digested, leaving just what you see on the screen. And so when you pull that down and sequence it, you get the fragments that were protected by some protein, and you can use that to infer the underlying structure. And one representation to get at this structure is something called the V-plot. And so it's a two-dimensional plot. On the x-axis, you have position of the fragment midpoint, and on the y-axis, you have the fragment length. And so if these are the fragments from these nucleosomes and the nucleosomes to the linker, project that down and make the plot, and you get something like this. And so again, y-axis, you get the fragment length. So for this, dual nucleosome fraction with the linker sequence, with the linker histone, you get a longer fragment with a midpoint right between them. And for the single nucleosome fragments, you get something at 147 base pairs-ish that corresponds to the position of the nucleosome. And here we're circling the dinucleosomes and the mononucleosomes. These V-plots are really, really useful in analyzing cell-free DNA because you can visually see some of the structure and then use machine learning methods to infer and predict the location of all of these different types of events. And so the difficulty with analyzing cell-free DNA versus something cleaner like ATAC-seq is that you don't have control over the different types of cells that go into the mixture. So with ATAC-seq, you can either microdissect, or you can put it through flow cell cytometry, or you can run single cell ATAC-seq, and then you get something that's very, very clean. But because cell-free DNA is all happening naturally, and cells are dying in different ratios, you get a mixture, and that's something that you need to deconvolve. So here's a cartoon of a few different types of cells, and these are all contributing, of course, to the cell-free DNA fraction. And so when you actually sequence a cell-free DNA and combine it, you get something like this. So this is our real data. So this is, I think, around 100 samples or so from WGS data that are all stacked on top of each other. And it doesn't look nearly as clean as something you'd get from like a CTCF side or a well-positioned nucleosome side from a ATAC-seq experiment, because again, it's all of these different features together. So this region actually has a TSS about right here, but it doesn't look like the sort of clean TSS that you get again from a housekeeping gene in ATAC-seq. You do see the nucleosomes. You see a nucleosome right here and a nucleosome right here, and you see the dual nucleosome fraction, but you also see a dual nucleosome fraction that's sitting over the transcription factor or the POL2 binding site. And of course, that's something that you wouldn't naturally see. So this is one of the central challenges when you're analyzing cell-free DNA, is to understand how we can identify these functional elements that are necessarily in a complex mixture. But we can. I mean, if you look at this, again, you see the nucleosomes, the dual nucleosome fraction. You see some of these cut sites that are moving down corresponding to places the nucleus is digested back. And you also see these small fractions that correspond to the POL2 binding events and the DNA pieces that are bound to that. So, so I'm going to walk through this example right here. So this is another example. And here we're looking at, again, WGS data that's centered around a TSS. And this is a good TSS because it's constitutively expressed across the hematopoietic cell line, which is the vast majority of cell-free DNA in a healthy individual. And you start to see a pattern that's very, very distinctive. So, at the top here, I have the V-plot. Again, x-axis is midpoint position, y-axis is fragment length. Over here, we have the fragment length distribution, basically the sum of this plot all the way across the fragment length axis. On the bottom, we have the smooth coverage. That's the sum across the bottom. And you see a couple of the things that I pointed out before. You see this mononucleosome fraction. So 147 base pairs is kind of a mean, although you get a little bit of additional DNA. You see the dinucleosome fraction. These are nucleosomes that are together, typically have a linker, and so you get the DNA piece intact. And you also get information about the TSS and the POL2 and transcription pieces. So in this case, the gene is being transcribed being transcribed 3' to 5'. And so going back, you see the TSS about right here that's represented by this big dip in the total read coverage and also represented in the V plot. You see the small fraction which corresponds to the POL2 complex and the DNA that's bound to it. And then you see the well-positioned nucleosomes that are around it. So again, TBP sitting about right here, you get POL2 binding, you get DNA bound to the POL2, which is protected from digestion. You see this completely accessible region right at the TSS that's very, very accessible and so is digested quickly. And the POL2 complex and all the pioneer factors that come in and bind here orient that first nucleosome about 147 base pairs downstream of the TSS. And so moving back and forth, again with the V plot, you see the structure, you can annotate these different functional elements, and you can get a sense for the underlying mechanisms here. And so again, here's the well-positioned flanking nucleosomes. Small fraction of the POL2 complex, the subnucleosome fraction, which is often active POL2 moving across the gene body, but also just normal degradation products. And then you see the cut sites. And this is where the name B-plot comes from the original Hennekoff paper, where you see this accessible region and then the regions that are being adjusted back to the edges of the protected regions. So there's a lot that you can do this besides TSSs. So another place that this can become useful is just looking at nucleosome positioning across a transcribed gene body. So the original paper that sort of discovered that we could analyze cell-free DNA fragments and understand some of the underlying epigenomic features was this paper from Snyder that was published in 2015. This is a figure from this paper where he's showing really well-positioned nucleosomes in a particular region, and then showing that you can do a Fourier decomposition on these to get the periodicity of the positioned nucleosomes. And this is useful because if you're looking at, a transcribed gene, you're going to have different epigenomic, different histone modifications that relate to the nucleosome positioning. And so by looking at that spacing, you can actually infer whether or not the gene is transcribed. Another thing that you can do with these, with the V-plots and the cell-free DNA is you can infer transcription factor binding sites. So here I have a CTCF binding site. This is actually target capture data. So in our product, we identify regions that are differentially regulated between the indication of interest and healthy individuals. And of course, one of the features here are transcription factors. They're very specific to different cellular contexts. And again, you can see pretty clearly in the V plots where all of these events are occurring. So this is a constitutive CTCF site. This is bound in basically all cells, which is why it's so clear. And annotating it, you see the small fraction that corresponds to the actual protein and then protected DNA that's bound to it. And then you see going up diagonal in the V plot, you see a longer fragment length that corresponds to a CTCF, bound or proximal to the nucleosome, just upstream of that. And in this plot, you can also see some of the cut sites that I was referring to earlier. So here you have a nucleosome, and you have digestion coming right back to the edge of it. And so that digestion goes to the single, the di, and the trinucleosome fractions all the way up. In both directions also, you get digestion from the left that includes the CTCF plus the nucleosome site. And so we can use things like these CTCF binding sites we have tools that can predict the location of these, predict differential location, and use that to infer, topological compartment boundaries and other more active regulatory transcription factors. Another thing that you can get from cell-free DNA is, of course, histone modifications. And there's a lot of information you can get just from the V-plots by looking at the total, the fragment length distribution. Something that's in H3K27AC is going to have a little bit of a longer fragment length than something that's bound up in chromatin and has the histone sort of keeping it tight. But we can also just run cell-free chip, and that's something that we've done here at Ravel. So as you guys know, there's different histone modifications that are associated with different active regulatory processes. You have H3K4ME3 that's associated with active transcription. You have H3K27AC, which is typically associated with enhancers, but is also often associated with active promoters. And you can see that in cell-free DNA. So there's a lot going on in this figure. So we ran cell-free chip using H3K4ME3, H3K27AC, and H3K9ME3. I'm showing a plot here for healthy cell-free DNA. So again, this is mostly hematopoietic in origin. And when you look at these different marks and compare them to samples you take from, PBMC, you get something that's very, very similar. You see striking H3K4ME3 peaks right over, gene starts and promoters. You see H3K27 enhancers and also along promoters, and you see the repressive marks in repressed regions. So we can also look at these different types of histone modifications across different annotated functional regions. So here we're looking at chromHMM annotations across the genome in blood cells. And then we're looking at enrichment across these different functional elements. And across TSSs, you see basically exactly what one would expect to see. You see H3K4ME3 flanking the TSS and the active promoter regions. You see H3K9ME3 with a little bit of depletion around the region. You see H3K27AC, again, that sort of mirrors the H3K4ME3 at the promoter site. But when you look at regions that are bound up in heterochromatin, you see, depletion of H3K4ME3, H3K27AC3, and H3K9ME3 heavily enriched in these regions. And so again, this is something that we run to understand and provide a training set to better figure out how these different accessibility patterns relate to the various histone modifications. But it's something that we actually can predict directly based on things like nucleosome positioning, spacing between nucleosomes, and other types of functional elements inside of the region. You can also use this, of course, for cell-free ChIP-6 cell type deconvolution. This is something that's been reported in the literature by a few different groups, both using methylation and cell-free ChIP-seq. We've reproduced this in-house, and you get something that's kind of what you expect and has been otherwise supported. We don't see much with brain or the things you wouldn't expect to see, luckily, in healthy individuals, but you see a lot of megakaryocytes, you see a lot of neutrophils, you see other cell types that are in the hematopoietic lineage. Although the pancreatic islets is something that we're still trying to figure out where that is. Maybe there's some cross-reactivity there. Of course, you also have methylation signatures. I don't know if this counts as chromatin, but it's a big enough part of the cell-free DNA literature that I thought it was worth discussing. So we've also run some 5MC assays to pull down differentially methylated regions. Here we have, different types of counts. The cool thing here is that, as everyone knows, when you look at cancer samples, you expect to see a lot more methylation. And sure enough, we see this when we look across stages. When you look at the healthy and the early stage, if you look at the early stage cancer, stage 1 and stage 2, you start to see a little bit of enrichment. But when you get to this metastatic sample, you see big enrichment of the stage 4 cancers with respect to the 5MC. And what's going on here is the tumor fraction in the cancer samples is of course, significantly higher. You get additional DNA methylation compared to the healthies. So that was a bit of a whirlwind, sort of moving through the different elements. Here at Ravel, so here at Ravel, we've developed tools that allow us to basically, trying to find a good example, do this type of annotation. So we look at target capture around differentially regulated regions, and then have tools that allow us to analyze the V-plots and predict all of these different functional elements. And so CTCF is an example, but there's also more specific and sensitive transcription factors, histone modifications, other types of functional elements. And we use this to predict the presence and to monitor different types of disease. And we've done our pilot study in breast cancer. And the reason that we focused on breast cancer was, well, frankly, partially commercial. Breast cancer screening already exists. And so it's easy to get access to samples, and there's an obvious product on the back end of that. But the other reason is that breast cancer detection has been pretty hard for a lot of groups that are using traditional techniques for disease detection. And we have a hypothesis that the reason for that is that breast cancer is a disease indication where most of the cell death is actually associated healthy cell death instead of tumor cell death. And that's really about the biology of breast cancer and breast tissue, that breast tissue is meant to expand and contract, and breast cells get out of the way for growing cells around them. So there's mechanisms by which cells have physical pressure put on them. They undergo apoptosis pathways. And so the early stages of breast cancer, most of the cell death is actually healthy cell death rather than cancer cell death. And this is something that people like me and Abyssal have been looking at for decades now. So here's a couple of plots, and this is from Fallon 2017, and then this is our own data in green that we've reproduced. And what you see when you compare healthy to breast cancer to colorectal cancer, and colorectal cancer being one of the disease indications that traditional techniques like DNA methylation tend to do pretty well on, is you see an increase in the total amount of cell-free DNA, about a 2x increase mean between healthy and breast cancer, and maybe 2.5x to colorectal cancer. So the total concentration isn't too different, but it's a significant increase, meaning that even in early stage breast cancer patients, you're seeing a lot of additional cell death. But when you look at the mutant allele fraction, which we're using as a proxy for the circulating tumor DNA fraction, you see something very different. In colorectal cancer, you see a mean of about 10%, whereas in breast cancer, you see a mean of about 2.2%. This again supports this hypothesis that in breast cancer, the majority of your cell death in early stages is healthy cell death. And so when we went to build an early breast cancer detection panel at Ravel, we had all these techniques for identifying these differential functional elements, and we focused our efforts on looking at breast tissue in general. So we said, can we find tumor-educated fibroblasts or adipose tissue that's associated with breast cancer and would give us additional predictive power? And so we identified regions that are differentially regulated between hematopoietic and breast tissue, and we built a target panel, and we applied our techniques for predicting all of these differential functional elements, and then put this all into a classifier. And we got something that that dramatically outperforms existing published results in the cell-free DNA space, and actually does significantly better than standard of care, which is the screening mammogram here. So what we're looking at here is the recall of our proof of concept study. So this is a little over 100 patients in the intended use population, which is women that regularly undergo screening mammogram. All the samples were prospectively collected, and we kept a held-out test set to evaluate our performance on. The red dots correspond to the point estimate. So this is the real results from our study, and the confidence bounds are bootstrap confidence bounds after doing some subsampling games to try and get a better sense of how this would generalize in the future. And when we fix our specificity at 89%, which is what the FDA fixes the mammogram specificity at, then we get recalls across the stages that significantly outperform the mammogram. We miss about half as many cancers in this pilot study. So I think this is a really strong piece of evidence that, by looking at some of these different functional events and from inferring the chromatin structure from the cell-free DNA, we're able to get at more subtle biological features. And we've applied these results in different settings. We have the ability to discriminate between different types of cancer. We can discriminate between positive and negative biopsies in breast cancer and have some ability to distinguish different breast cancer subtypes like ER positive, et cetera. So here's the team. Anshul was my postdoc advisor. I think he spoke at the last one. He's been a great friend and a mentor for a long time, and we've been working on these methods together. Paul's my co-founder. He invented a TAC-seq and founded a company, Epigenomics, and then moved to Ravel so that we could apply some of this work in different settings. And here's the rest of the team. Social shout out to Omid and Arthur, who actually put together all of my slides for me. This talk actually ended up coinciding with us closing a funding round. So it's been a crazy week, and Arthur and Omid really came through to put this all together. So thank you guys very much. And yeah, I'd love to answer some questions.
Wonderful. Thank you so much, Nathan, for this great overview and the great talk.